There are many factors that affect our health. These are the genes that you are born with, and then there is the interaction that takes place between your genes and the environment from the moment you are born. We have managed to map the human DNA and work out which of the genes can cause diseases. But the research about which environmental factors affect gene expression is still limited. Studies have shown that it's not just one environmental factor affecting one gene that causes disease, but it's rather many environmental factors causing many genes to be switched on or off that predicts disease. We are each born with an individual genotype or genome, which is the set of genes in our DNA or a complete DNA sequence, which is responsible for a specific trait and which makes a person completely individual. Early life exposures of maternal intake of fat, protein, and total energy intake, and gene-environment interactions seems to indicate regulation of specific genes, resulting in development or avoidance of disease in later life. Human longevity can be explained by our genes only 25% of the time, whereas lifestyle habits such as exercise, nutrition, and stresses contribute 50%. Population traits also need to be taken into account. For example, among a smoking population, people that have a gene that is turned on by smoking will most certainly obtain that disease, whereas people that don't live in that smoking population but still have that gene won't have the same risk of disease development. While genetic research has been successful in predicting how certain nutrients, vitamins, and specific environmental factors will affect genes, studies have been less successful in predicting which individuals in a population will acquire certain diseases. Also, large-scale studies have only served to establish that it is not individual genes that perpetrate disease acquisition, but it is many hundreds of genes that work together to create chronic disease. A combination of exercise, nutritional requirements, and stress relief techniques also all play a part in how the body reacts to change gene expression, but also how it reacts to certain other genes. Examples of that are the changes physiologically in reaction to the relaxation response. Studies have shown that certain people are genetically predispositioned to react differently to stressful stimuli, leading to chemical reactions and gene expressions, as well as choices in lifestyle behaviours. This is also true when it comes to genetic reactions to environmental pollutants. Some gene variations can have negative or more severe reactions to certain pollutants, leading to disease. Also, variations can occur due to different personalities, whereas someone who is more adventurous will take more risks and create different neural pathways, which can take them along different life paths while also affecting gene expression. The way we experience the environment we live in is very individual and very much shaped by our thoughts and feelings. Methylation can be described as the adding of methyl groups to DNA, which marks those genes to be either activated or silenced during transcription. Diets high in folate and B vitamins are methyl-containing nutrients that leave epigenetic marks. Also, folate is an important nutrient as it can stimulate DNA repair. Neuronal activity can modify their pattern of DNA methylation in response to physiological and environmental stimuli. The precise regulation of DNA methylation is essential for normal cognitive function. When DNA methylation is altered as a result of developmental mutations or environmental risk factors, such as drug exposure and neural injury, mental impairment is a common side effect. The investigation into DNA methylation continues to show a rich and complex picture about epigenetic gene regulation in the central nervous system and provides possible therapeutic targets for the treatment of neuropsychiatric disorders. Histone modification can also affect gene expression and therefore activation. DNA methylation works with histone modifications and microRNA to regulate transcription. Histone proteins can help to package the long strands of DNA into the small nuclear compartments. Chemical modifications influence not only how DNA strands are packaged, but also their transcriptional activity. Histone modifications that loosen DNA association with histones generally provide a permissive environment for transcription, whereas histone modifications that tightly package DNA and histones repress gene expression. DNA modification and epigenetic processes such as methylation and modification of histones can affect behavior responses to environmental factors. These processes determine individuals' levels of resilience and how stress is dealt with. This personalized response to the activation of the sympathetic system, fight or flight, can be triggered in isolated or lonely people, as well as people who experience high levels of stress at work. These heightened reactions can leave stress-sensitive people open to type 2 diabetes and depression. The epigenome are the chemical compounds and proteins that attach to the DNA, which act like a switch to turn each gene on or off. The epigenome can be inherited and also altered by environmental factors. Both changes in the genome and the epigenome can cause cancer by switching on or off genes that are responsible for immune response or cell growth. Epigenetics is central to genome structure and function. It is clear that both genetics and our environment play a major part in how our genes are expressed, and both factor in to our individual risk of chronic disease. The World Health Organization has reported that nearly half of global mortality is due to environmental exposure, such as cigarette smoking, air pollution, alcohol use, and high levels of salt consumption. It has been found that the above exposures are needed along with a certain genetic background to explain chronic disease developments. Although due to the ease of study of genes when compared to environmental elements, which are often self-reported in studies, gene expression is often shown to be the reasoning behind developments of cases of chronic disease. That's why further studies need to be conducted which take into account specific environmental factors in the creation of chronic disease. For years, scientists have argued of the importance of nature or nurture, but now there seems to be a third factor that can influence a person's genetic expression. Through the use of twin studies, it has been shown that individual twins can be different due to early developmental mutations occurring. This can occur by methyl groups being added to certain stretches of DNA, either turning on or off a particular gene. The environment in which we live in has a profound effect on our gene expression, which in turn shapes our lifestyle behaviours and therefore our health. Furthermore, epigenetic differences have been shown not to be just due to genetic makeup or the environment we live in, but has a certain element of randomness to patterns of methylation in an individual. This could be evolution's way of ensuring survival by creating random epigenetic changes, which creates more variations in gene expression and more chances of survival, in a constantly changing environment. This can be seen in Charles Darwin's book On the Origin of Species, where he attributed importance to random, undirected change that provides material for natural selection. To be able to deliver accurate, personalised lifestyle advice, accurate measurements need to be taken of not only our genetic makeup, but certain elements that can show in blood samples, such as C-reactive protein, liver enzyme markers, and homocysteine levels. These factors can show direct or indirect causes of not only symptoms, but a person's gene profile, so creating a fuller picture of all underlying causes of disease. By having this information in patients' files, preventative measures can be taken in regard to lifestyle recommendations to counteract any abnormalities in their test results. Certain genetic profiles can show that certain people have low tolerance to environmental exposures and drug reactions, which is helpful when making recommendations for preventative health measures. Genomic medicine is useful in that it can predict the pathway of a disease may take when taking the genetics of a person into consideration. Also, which medicines will be of benefit to a person can be determined by genetic background. The maintenance of health or the development of disease is all controlled by environmental factors such as nutritional intake, exercise, environmental pollutants, and stress, and that epigenetically alters our gene expressions. We are born with the genes that we inherited from our parents that predisposed us to susceptibility to disease or good health. The way that we use those genes or the effect we as individuals have on our genes is up to us. We can create health or we can create disease. Thank you very much.